Dear students, in the series of basic courses, we will continue the basic course of immunology. And today's lecture, we will discuss about the subsets and function of helper T cells. So first, we will see at the end of this lecture what you will be able to learn. So how helper T cells are differentiated into multiple subtypes and what are the function of these subtypes. Why such diverse type of helper T cell is needed by our immune system. And at the end of this lecture, you will able to learn how TH1 cells, TH2 cells, and TH17 cells and T regulatory cells work to eradicate the infection. So before starting our next lecture, let's recall the few points from our previous lectures, what we have learned so far. So we know that activated CD4 positive T cell become the helper T cells. That is the CD4 positive T cells, and then they got converted into the T cell that is called the helper T cells. So after understanding that activated CD4 positive T cells form helper T cells, there are two questions that come to our mind. So first, why they are called helper T cells? So why, as the name is indicated that because they are involved in helping the other immune cells, so that's why they are called the helper T cells. It means why they called as a helper T cells or why they given this name, because they help the other cell of the immune system to eliminate the pathogens. So that's why they are called the helper T cells. And the most important, they are the hero of the immune system. So they perform a lot of functions. They are triggering the many of the immune system cells. So why and the another questions, and what are the functions, and it means how they trigger the immune system, how they help the other cells of the immune system. So the next question is, what are the function of these helper T cells? So today's lecture will give you the answer to these questions. And if you look for here, so these T cells actually, in order to understand the function of helper T cells, what we have to done that we need to develop the understanding of subset of helper T cell, because for their functioning, they first got differentiated into the different subtypes and then they perform the functions accordingly. So therefore, if we want to understand the function of helper T cells, we have to understand the subtypes of helper T cells. So in this lectures, we will discuss the most common four subtypes like TH1 cells, TH2 cells, TH17 cells, and T regulatory cells. So what are the basis of their classification of the helper T cells? So they are classified on the basis of the cytokines that induce their differentiation, on the cytokines they produce and secrete, and the master transcriptional regulators. And then why we have given the name like TH1 cells and TH2 and TH17s and T regulatory cells. So basically the first two, if you look for here, the TH1 cells and TH2 cells, actually they are given the name like TH1 cell. These are the subtype of the helper T cells which are discovered for the first time. So on the basis of their discovery, they are called the TH1 cells. And then the second was discovered another subtype that is called the TH2 cells. Then but why we call that not the third cells, why we call the 17? So that we will discuss in our coming slides, why we call them the TH17 cells and why they are called the T regulatory cells. So we are going to discuss in our coming slides. So in this lectures, we will discuss the four main type helper T cells, which we have mentioned already. And these helper T cells differ from each other in the several manners. Like as we know that they produce a different kind of cytokines, and they even differentiated by the different kind of the cytokines, and even they are regulated by certain kind of the transcriptional regulators. So why such? The first question before starting the subtypes. So let's try to answer this question. Okay, why such diverse type of helper T cell is needed by our immune system? So maybe the answer of this question is slightly and easily to understandable, because we know that we have millions of different kind of pathogens that could be the different kind of bacteria, viruses, even several kind of the parasites, lot of the toxin materials. So because of we have the different type of pathogens and infectious agents, so we need the different type of subsets of helper T cells. And then other is they also have a different target sites and they have a different target cells. Let's try to understand with the help of few examples. So if you look for here, we have a mycobacteria. That is a kind of the pathogens. Actually, they resist killing by macrophage. And then if we talk about another example, that is a helminthes. So helminthes, they are the parasites, but they are too large to be phagocytized. So how we can cope with such diverse type of the pathogens like bacteria, helminthes, and many more. So in that, that's why we need a diverse type of the cell by our immune system. So that is the that we slightly maybe we could understand that okay, why we need the diverse type of cells by our immune system, because they have a diverse type of pathogens, infectious agents, different target sites, and different target cells. So let's start to understand next one in the subtypes of these cells. So if you first into the subsites of what we are going to talk, first we will try to understand how these subtypes led to discovered and how their discovery could be possible, and what led to the discovery of TH1 and TH2 subsets. So if we take the examples here, immune response to mycobacteria. For example, we already mentioned the example of mycobacteria and helminthes. So by taking the same example, we know that the mycobacteria and helminthes, they are the different kind of pathogens. And if we talk about the CD4 cells, in both cases, they are required by the to against the response of this kind of the cells. So now let's see if you can see here, if the foreign pathogens which entering into the body, for example, in this mice, that is the mycobacteria, and then we have another kind of pathogen that is called the parasites, and in this case, there's the helminthes. So then how actually in both cases the CD4 T cells give the response, but how they give the response, that is important to understand. So immune response in the mice infected with bacteria and helminthes are very different. So if you look for here, mycobacterium infection induces the macrophage activation. So this is when when the mycobacteria enter in the body that infected the any animals. What happened? The CDT4s lead to the activation of macrophage. So macrophage activated, and as a result, what they does, they also release the certain kind of factor, that is the IgG. But if we talk about in terms of helminthes, that is the parasites, so while helminthes infections recruited eosinophils along with the production of, if you can see here, IgE. So it means they are giving the different responses. So is of course, here are the questions, how can the same CD4 positive T cell trigger such distinct reactions? So let's try to understand how this could be possible. So for this purpose, actually, we can also hypothesize that maybe the CD4 T cell consists of the subpopulations that mediate the different response. This is our common observations, or maybe we even can easily hypothesize that because they are giving the different responses. So it means they have this maybe inside within this CD4 positive T cells, they are giving a sign kind of different kind of population. So a certain population responding to the helminthes and certain responding to the infection of the mycobacteria. So which forces are behind this differentiation expression and functioning the helper T cells? So if we talk about that, based on this hypothesis, in 1986, groundbreaking study led to the identification of mouse CD4 positive TH1 and TH2 clones based upon the differentiation production of cytokines. So it means, and that as you can see here, they reported their studies in this paper in 1986. So that led to the discovery of these two subtypes, TH1 and TH2. So they discovered the first subset, that's the TH1, and that's given the name as the TH1, and then they discovered the TH2 subsets. So of course, we understand how they discovered the TH1 and TH2. So let's try to understand how the discovery of TH17 subsets. So some inflammatory diseases thought to be caused by TH1 cells were not prevented by eliminating the TH1 cells or their cytokines. As you can see the example here, let's suppose this is a mouse model, and what we know that they can induce a certain kind of inflammatory disease. But who is responsible for this disease? That is the TH1. So it means what we call it as a TH1 mediated inflammation. And if we want, this inflammation should not be called. So what should happen? If we will taken out, we will eliminate these TH1 cells and their even cytokines, so that disease should not happen. But what happened? Still that inflammation persistent. So it means that there is something else other than the TH1 cells that also take part in that kind of inflammation. So that one actually, this response, and that was not completely mediated by the TH1 cells, or might be there is some other subpopulation of the T helper cells. So that other population was actually, if we see for here, that led to the discovery of another kind of the subset of the helper T cells, and that's we given the name as a TH17 subsets, and that was discovered in 2005. So why we are called them as a weird name, like instead of why we are not calling as a TH3 cells, why we are calling the TH17 cells? So that's we are going to learn in our coming slides. So why we call them as a TH17 subsets of the helper T cells, and that paper that was reported in the Nature Immunology. So now let's start how helper T cell works. Of course, now we understand that helper T cells, firstly, what they do, they differentiate it into the various subtypes, which we have already discussed. Now we will take how actually each subtypes works. This is a journal as a whole for the helper T cells. So helper T cells work by secreting the cytokines, which communicate with other cells of the immune system and activate them to eliminate the pathogens by an appropriate immune response. So we know that there is a certain kind of cytokines, but how helper T cells are differentiated into the multiple subtypes, and what are the function of these subtypes. So let's start with the first one, that is the TH1 subtypes. So if we talk about with the TH1 subtypes, so we will try to develop an understanding of the TS and TH1 cells, or we call it as a type 1 helper T cells. So when they work in case of intracellular infection. So one thing what we are getting here is that each subtypes will give the response to the different kind of pathogens. For example, some will be response to parasites, some will respond to fungus, some will respond to the extracellular infection, some will respond to the intracellular infections. So if we talk about the TH1 cells, what they respond in case of intracellular infections. That's the process we have already discussed. Is somehow is similar as the cases APCs in antigen presenting cells. They present the foreign antigens to present it to whom? To the helper T cells. Now these helper T cells, before functioning, what they do? We have a certain kind of cytokines. They send the signals to these helper cells. We have intracellular infections. So then what they do? Macrophage presenting the peptide of intracellular pathogens, and then they start secreting IL, what we call it, interleukin 12. And then that interleukin 10, once antigen is recognized by the T cells, and T cells got activated. And now what happened? And then that activated T cells, they release and by macrophage and regulate the TH1 differentiation. And how they regulate the TH1 differentiation? Actually, they start to activate the transcriptional factor. And but in case of the TH1, which transcriptional factor got activated? That is the STAT4. So what they do actually, IL-12 regulate the TH1 differentiation through the activation of STAT4. And now as a result, that's why. And now they got differentiated into the type 1 helper T cells. And now these one, what they will do, they release the certain kind of the cytokines. As you can see here, they release IF and gamma, and this IF and gamma activate the macrophages and increase their microbicidal activity. And this IF and gamma also stimulate the production of antibodies to promote the phagocytosis by the neutrophils. So this is how type 1 helper T cell works. So if you look for the other things is also secreted by these, this is a interleukin 2. So what actually it does? It does the interleukin 2, the production of interleukin 2 enhance the growth and differentiation of T cells. So as a result, further T cells got differentiated when they know that they have a kind of intracellular infections. So now we have got the understanding of how the TH1 subcell works. Let's try to talk about the case of what TH2 cells, how the TH2 cells works. So basically, actually, the works in case of infection by parasites such as the helminthes. And what they does, actually, if you look for here, when the antigen presenting to the helper T cells and they start to secrete a certain kind of cytokines, and in case of TH2 cells, the cytokine is interleukin 4. And that interleukin 4 released by the mast cells in case of infection by helminthes, and that regulate the TH2 differentiation. And again, as we learned in the last case, what they do, they involve the transcriptional activity of the state 4. But if we talk about in terms of type 2 helper T cells, so they activate the transcription factor GATA3. So they got activated, and now these cells will be got differentiated into the TH2 cells. And IL4, what we can say that IL4 regulates TH2 differentiation through the activation of GATA3. But if we talk about the first one, we say that IL12 regulate the TH1 differentiation through the activation of state 4. So these are, even we can compare later on which type of subsets requiring what kind of cytokines and what kind of the transcription factors. So that is important to memorize if you want to understand how these subsets works actually. And when they got activated, after that, what they do, under the effects of GATA3, TH2 cells start secreting the various kind of cytokines. And actually they secrete the 3 different kind of cytokines. As you can see here, they secrete the IL4, IL5, and another kind of the cytokine that is called IL13. So let's look what's the function performed by these cytokines. So if you look for here, for the IL4, it promotes the production of IgE antibodies. What does by the IL5? So IL5 activate the mast cells and eosinophil. That means they perform anti-parasitic activity. And then if we look for here, the other type that's called IL13, it induces the production of mucus. So these are the important functions performed by the TH2 helper T cells. But let's try to understand how, because there is one thing very interesting in that, but we are trying to learn that we mentioned earlier these helminthes could not be phagocytized. If they could not be phagocytized, then how they are presenting, how they engulf it, and how they are presenting that antigens to the helper T cells. Let's try to understand and how our immune system eliminate the helminthes from our body. So let's try to understand that phenomenon. So if you look for here, how dendritic cells process and present the helminth antigens to the T cells. And then we know that the micropathogens could not be phagocytized. If they could not be phagocytized, so we have to scratch our head. So how they works in that case? So if we actually for here, so nature always have the other options. Nature always provide the, of course, the many options for our betterment. So in that case, what actually they does, if we talk about the helminthes, they are the micropathogens, a condition prevent from being ingested by the phagocytes. So then how the dendritic cell present the antigens to the T cells. So if we talk about that one, actually, and how these dendritic cell present their antigen to the T cell during the infection by helminthes, the product secreted from the adult worm intestinal and uterine contents during the egg depositing that play a fundamental role as a modulator of phagocytes activation. So what they do, these molecules such as protease enzyme and glycolytic enzyme, what they does is, you can see here, they are secreted from here, and they are called excretory or secretory antigens. And now these antigens can be taken by the cells. When they take these antigens, and now they are picked up by the dendritic cells, and then they are displayed as the non-cells on their surfaces, what we have seen earlier. And then our cells and helper T cells got activated, and then helper T cells give the response to kill that kind of the pathogens. So how like IL-5, IL-13 and other working in that phenomena. So let's try to understand that one. Okay, how these cytokines contribute to the elimination of helminthes. So what they produce, we know that they have a Ig antibody. So what Ig antibody does? So Ig antibodies coat the parasite surface. And then what happened? Why they coat the parasite surface? Because the mast cells and eosinophils have a receptor for Ig on their surfaces. And then these enzymes released by the eosinophil and mast cell penetrate the parasites to kill them. And then they are inside the parasites. And now, what the function of mucus? Why the mucus was secreted in that case? So if you look for here, we have a mast cells and we have a eosinophils. And then after that, let's try to understand what the mucus does actually. So mucus promote the expulsion of parasites from the gut. So in that way, they release a certain kind of enzymes, and then these enzymes actually digest the, or what we call them, kill that kind of pathogens, and that pathogens eliminated from the body. And then how actually TH2 cells are to kill the pathogens. So let's try it. So far we have learned the TH1 cells and TH2 cells. So let's try to understand how TH17 cells works and why we have that weird name instead of TH3 cells. So let's try to understand when TH17 cell works. So they work in case of infection by fungus and extracellular bacteria. And similarly, we have know that how they will be presented. In that case, first of all, of course, pathogen will be engulfed by the immune cells, and then they will be by the APC, they will be presented via MSC molecules to the helper T cells, and then these macrophages release a certain kind of cytokines. If we look for here, they will release IL-6, interleukin 6, and also another factor that is called TGF beta. So TGF beta and IL-leukin sector releases, and then they activate the transcriptional factor. In this case is a state 3. So in the first case was a stat 4, then got a 3, and now is a stat 3. So stat 3 is a very important, and even state family is a very important in terms of the immune system. So then as a result, these cells got differentiated into the other kind of subsets that is called TH17 subsets. So what happening actually here, if you can summarize here, IL-6 regulates TH17 differentiation through the activation of state 3. So we have learned so far who differentiate, what kind of cytokine is required for the TH1, TH2 and TH17. So if you talk about the TH17, which cytokine is required? That is the IL-6. And it does the differentiation of TH17 cells. How it does the differentiation? Via activation of state 3. So that's what we have learned that. And after the activation of state 3, so of course, they will release certain kind of cytokines. And then of course, what they will do, these cytokines will work, as in this case, you know that is the releasing cytokine is the IL-17, and there's another cytokine, this is the IL-12. So that cytokine, because they find out the other kind of the subsets, and then they found that they release interleukin 17. So that's why they given the name as a TH17 cells. So now we'll try to understand what the function is done by these cytokines like interleukin 22 and interleukin 17. So let's try to understand the function of these cytokines. So IL-17 recruits the further neutrophils to site of infection to phagocytose the invading pathogens, that in this case is the fungus and extracellular bacteria. And what the function does by the IL-22? So IL-22 stimulates the epithelial cell to produce antimicrobial peptides that resist the microbial invasion. So these are the both how and what they do and how the TH17 cells works. So next talk about the last subsets of the helper T cells, that is the T regulatory cells. And These are the very important sets of the, of course, we mentioned that the T cells are the hero of the immune system. And among them, the regulatory are the very important subset of the helper T cells, because they also now we understand then what happened when the like intercellular pathogens enter, when the parasites enters, and for example, when the fungus or maybe the extracellular bacteria enters. But if these pathogens are eradicated, so then how our immune systems be come back and suppressed, so it does not damage to our own body cells. So that are the cells that are called T regulatory cells, and because they are involving in the regulation of immune systems, so that's why they are called regulatory T cells, and they're also called suppressor T cells because they involved in the suppression of immune system. So when work, they work when the pathogen is eradicated. So they limit the tissue damage, they suppress and limit the immune response and prevent the autoimmunity. So this is very important sets of subsets of helper T cells, and they perform very important function in our body. So how they actually differentiated, it means how helper T cells got differentiated into the T regulatory cells. So if we talk about that, what happened actually, if you look for here, in case of regulatory T cells, so they are differentiated by the an important factors here, and they release the interleukin 10 and TGF beta. And interleukin 10 is a potent suppressor of macrophage function, and while TGF beta induces the production of T regulatory cells, so they will produce more number of T regulatory cells. So what they can do, so as a result, then they can protect our they can protect our body from the attack of immune cells. So when the work, what we have learned so far, we say that the work when pathogen is eradicated, and T regulatory cell communicate with other cells of the immune system and help them to stop their effector function. So that's what we have understand about the T regulatory cells. And then once formed T regulatory cells are secreting IL-10 and TGF beta, which helps to inhibit the inflammation and immune response. And we also discussed what is the role of IL-10, and we have discussed what is the function of TGF beta in this case. So as they play an important role in the regulation of immune response, so that's why they are called regulatory T cells. So let's try to understand how helper T cells are differentiated into T regulatory cells. So that is in the, like we have learned how they got differentiated into the TH1, TH2 and TH17. So let's try to understand how they got differentiated into the T regulatory cells. So T regulatory cells expresses high level of transcriptional factor, but in this case, the first case was a STAT4, COTA3, STAT3, but in this case is a FOXP3. So what they do is they express the high level of transcription factor that is called FOXP3, and which serve as their master transcriptional regulator. And then once they're like, if we know that when they are got differentiated from the helper T cells into the T regulatory cells, then other questions come to our mind: how they identify of the T, how identity there, and how they maintain. They should kept like as a T regulatory cells, and then they should not be behave like their parental helper T cells. So who maintain this phenomenon? So they maintain by the high level of FOX division throughout the life of T regulatory cells. And then these T regulatory cells express the high level of transcriptional factor of FOXP3, and this FOXP3 serve as a T regulatory cell lineage master transcriptional regulator. So that's what we have learned so far. So let's try to summarize our whole today's lecture. So what we have learned so far, we have started with the terms that helper T cells have several subtypes, but we have discussed here their most common subsets, and that we call them as the TH1 cells, TH2 cells, TH17 cells, and lastly we discussed the T regulatory cells. Then we also discussed one by one how they got differentiated and what function they perform. So if we talk about the TH1 cells, TH1 works against intracellular infection by activating macrophages and stimulating the production of antibodies. What does how and what function performed by TH2. So TH2 work against parasitic infection by activating the production of antibodies and stimulating the mast cells and eosinophils. And if we talk about the TH17 cells, so TH17 work against the fungal infection by activating the neutrophils and production of antimicrobial peptides. These 2 points we have discussed earlier. And if we talk about the T regulatory cells, the most important subsets of the T helper cells, so they work when the pathogen is eradicated. What they work by suppressing the immune response. So it means they actually prevent the autoimmunity. So that is all for today. So what we have discussed here. So let's see what we are going to learn in our next lecture. So in the next lecture, we will discuss how there are the 2 subsets of T cells, helper T cells and cytotoxic T cells. So in today's lecture, we have discussed the helper T cells. So in the next lecture, we will try to discuss how cytotoxic T cell works. So that's all for today. If you like our lecture, so please subscribe to our channels, not only for the conceptual biological lecture, but also much more. It means in this channel, we will also going to start a few other kind of series like career counseling series, and we more importantly, we also going to start the scholarship guidelines, especially for Chinese scholarships and for the scholarship Japanese scholarships. So if you're interested in those ones, so please subscribe our channel today. Thank you all, and thank you for watching our this lectures.